Hi, good day, and welcome to the Ministry of Agriculture's Research Division's presentation on citrus greening. It is commonly called Huang Longbing or citrus greening disease. My name is Julia Paris, and I'll be the plant pathologist attached to Research Division in the Ministry of Agriculture, and I will be running you through this disease. Now, before we start, I just wanted to mention that there are different types of surveys. So when we are doing a survey for a disease, we can do something called a detection survey, which is to identify if the disease is present or absent in the country. So on previous presentations, we would have talked about frosty pod. My colleague, Mr. Kishore Ragbe, had talked about frosty pod, and my other colleague, Mr. Terence Jack, talked about citrus canker. Those are diseases that are not in Trinidad, so hence we are doing a detection survey to determine if it's present or not. We also have what we would call a monitoring survey, which is just to identify the characteristics of the pest population, and that would be what we would do for the disease citrus greening, because it's already been declared in Trinidad and Tobago. And of course, there's something called a delimiting survey. So if we pick up a new disease, we have to identify and establish the spread of that disease. So that is when we do a delimiting survey. Now, why am I talking about this? Notifiable pests and diseases is the responsibility of the MPPOs or the Ministry of Agriculture. We have international reporting obligations to declare that a specific pest or disease is in your country. And the only way we can do that is surveillance and with the help of the general public with reporting these pests. So it is also in our Plant Protection Act, citrus leprosis and citrus canker. As we are talking on citrus, these are two diseases, citrus leprosis. My colleague would have done a presentation on citrus canker. So it's in the act that it's a notifiable disease. So you must report if you've seen this disease or if you think you've seen this disease for us to go and do further looks. So the topic we're on today is HLB or citrus greening. What is citrus greening? Citrus greening or Huang Longbing is a disease that is vectored by a psyllid called Diaphragma citrui. But the causative agent is a bacterium, is Candidatus liberatica, and the strain that's in Trinidad and Tobago is Asiatica. Now, this disease has been declared in the Caribbean and in various countries such as Barbados, the Dominican Republic, Jamaica, and Trinidad and Tobago. This is just a global distribution of this disease. But as I said before, there are different strains. So in the orangish color here in our side in Central America, Bolivia, so forth, that is the Asiatica strain. And then in the African strain is the Africana strain. And so this is just the distribution. There are different strains, but the most prominent is the Asiatica. The Asiatica, the Americanas, and the Africanas. So why is this important? So why are we monitoring for citrus greening even though we have it? So first of all, as I said, citrus greening is a bacterial disease that is vectored by a psyllid called Diaphragma citrui. So we have to monitor the vector population with the use of sticky traps or sweep nets. So just for instance, the yellow sticky traps can be placed as 2 meters, preferably around citrus orchards. So as I said, it's a psyllid that directly feeds on the flush or the new growth of citrus trees. So the population can be monitored around when we have new flushes for citrus, so usually in the rainy season. However, when they feed on these shoots, the shoots turn black and may cause the leaves to twist. So if you are looking out for the psyllid or citrus greening on a whole to see if your plant is you look at the new flush and carefully look at the new flush. So how the psyllid works, it feeds on the flush, and if the tree itself is infected, the bacteria will be will be ingested by the psyllid, and when he feeds on another tree, another healthy tree, it will inject the bacteria into the healthy tree. So this is just a picture of the citrus psyllid, but it's very zoomed up. So I will go into the, so this is how it really looks. It just looks very young psyllids on the tree. As I said before, it causes a twisting of the new flush, because you can see this is the flush. And so when you're watching it, it's very small. So sometimes you use something called a tap test. So what are the symptoms to the trees? So symptoms may appear anywhere months to years after the infection, but there are some main symptoms. So you have one is asymmetrical chlorosis, corky veins, and irregular peel color. So let's go with the asymmetric or blotchy mottle. It basically means if it's yellowing on one side, it's not evenly yellowed on the other side. I notice, so I hope you can see from the picture that you have one side kind of green and the other side kind of yellow. So that is one symptom of HLB in citrus trees. Another symptom is these raised corky veins. So as you can see from the picture, the veins are quite raised, and you can see some instances of the asymmetrical blotching. The other symptom is the fruit color inversion. It ripens from the top, so you'll have premature drop of fruit and poor quality of fruit. So the yellowing is not ripening how it's supposed to be. And of course, we have internal symptoms. So when you cut the fruit, it's distorted like this. So the fruit is curved inside like that. You also have what we would call aborted seeds. So the seeds are no longer usable, and the reason that is such a problem is because seeds are usually one of the main methods for obtaining rootstock for citrus. So what I wanted to stress here is that in picture A and B, if you can look carefully at A, it looks like a healthy tree, but it's actually infected with the HLB disease. Because, and even if you look at the left, my right, I don't know how it's coming across on your screen, the leaves on each side, it's very hard sometimes to tell healthy leaves from asymptomatic HLB leaves. And of course, well, the symptomatic leaves are easier to detect. So your tree can look healthy, but it can be infected with the HLB disease. So why are we monitoring? So what is the monitoring program for citrus greening disease? Right? So the citrus greening disease was detected and confirmed in 2017. And we have conducted 2 island-wide surveys, one in 2017 and one in 2021. So what was the strategy implemented? The ministry implemented a HLB committee, and the management strategy was to provide clean, tolerant planting material, provide good agronomic practices, psyllid management, improve testing through PCR, monitoring programs of farmers' field, and education of farmers and extension. So this is one of our education programs for the general public. So as I said, management of HLB. So surveys conducted using iodine tests. Now, the iodine test is a test because one of the symptoms of HLB is that it would develop abnormal amounts of sugar in the leaves. So the iodine test is what we would call a pre-screening methodology. It is not a confirmatory test because there are some citrus varieties that can store some sugar. So the detection test is what we will use PCR technology for. So also, it is also a vector disease, and that is very important in the management of the disease. So even though we had detected the disease in Trinidad, we also had to make sure that the vector, which is the transporter agent, is also detected in Trinidad. And one of the things, we imported tolerant rootstock material and varieties, rootstock material. So the operational procedures. So the ministry conducted through the research division, my colleague, Mr. Terence Jack, and Mr. Kishore Agbe, they spearheaded this part of the project, where they would have visited the greenhouses in AST, where we would, and I forgot our colleagues in entomology, Mr. Amil Bash and Numala Gayasing. They would have gone to the greenhouse and they would have installed sticky traps to determine if the area that these clean materials are growing has any insect for cause of concern, which is Diaphragma citrui. We monitored for any disease, and visible observations were made monthly. Any samples collected were taken to the plant pathology lab and confirmed with testing through HLB at the biotechnology lab at the Ministry of Agriculture. Now, these are some of the pictures since 2019, where we've been monitoring the planting at AST. So why, you ask, are we doing PCR for a plant disease? So HLB, which is Candidatus liberatica asiatica, is a bacteria. However, it cannot be cultured through standard means. It must be detected through PCR because it cannot be cultured by the lab. So in order to reach to that point, the ministry had to improve or develop a PCR laboratory in order to conduct accurate testing. As I said before, the iodine test is good as a pre-screening test, but the PCR is the confirmatory test used by international laboratory. We can also send samples to international laboratory for proper confirmatory testing, but in a monitoring system, it is more cost-effective and practical that your MPP or your Ministry of Agriculture has a PCR testing facility in order to detect if this test is present. Now, I will go more into that. Reason why is because, as I said before, plants could look healthy but actually be infected with this disease. And two, the disease has a sorry. So I'll go into the PCR testing. So PCR testing. So what we will do, we would collect the sample from the farmer's field, and then we would do DNA extraction from plant tissue, and then we would run PCR analysis, and the results is recorded, and then the results is provided to the stakeholder. We also send samples to CABI for an international accredited lab for testing. But as I said before, we've already declared that this disease is in Trinidad and Tobago. So routine testing for monitoring surveying is done internally. The reason why we also do PCR is because there are a lot of nutritional deficiencies that resemble HLB, but are not quite HLB. And as I said before, even though iodine is used as a good pre-screening methodology, the only confirmatory test is PCR technology. So we have a lack of nitrogen. I don't know if you're seeing the pictures clearly, but the older leaves the older leaves turn yellow first. So it's like a very lightish green color, and mature leaves are bleachy. So as you can see from the picture, it's not the asymmetric mottling, but it's a very lightish color. So when you watch the tree looks yellowish, but instead of being HLB, it can be a nutritional deficiency in nitrogen. Similarly with potassium, the older leaves, it occurs on the older leaves first, and the yellowing of the tips and margin. And of course, you may have smaller fruit size. You also have magnesium from the picture. It occurs on the mature leaves, and there's an inverted V shape surrounded by the yellowing. There's also sulfur, which is occurs on, is it like that same pale yellow looking, and even iron, as I said before, but iron, however, occurs on the younger leaves with a very distinct green vein and a yellowish to white color. Then there's zinc. It occurs on new growth and it reduces leaf size, and there's a kind of a mottling effect. So zinc is one of those that looks like HLB, but once you've seen it a couple times, you know it's not, because it has this very green ridge, right? So these are just, so I went back again just to show you all. These are just some of the reasons why HLB is hard to detect, because there are a lot of nutritional deficiencies and even some diseases that resemble it. So as I said before, we don't currently have these diseases in Trinidad, but as I said, it is a notifiable pest. My colleague, Mr. Jack, would have done one on canker, and this is just some pictures on citrus leprosis, right? As you can see from the leaves, it's a very distinct shape, even the stem and the fruit. So take a good look, and if you see this, or if you think you see something similar, at the end of the presentation, there will be an email address where you can send a picture to report, and we will follow up. As I said before, this is citrus canker. We would have done an entire presentation on citrus canker. So this is just a review, just for you all to get the understanding that how these diseases interact with each other. So what are our recommendations? As I said before, this citrus greening, or HLB or Huang Longbing, is a vectored pathogen. So of course we must try to control the psyllid populations. However, we have made some observation, but not I, the entomology unit, Mr. Amelbash, has made some that there are a lot of natural enemies present, and that the psyllid population is not as high. So we would recommend the use of insecticides if you think your orchard is infected with citrus greening, and around this time where citrus may be flushing due to the rainfall, spraying to reduce the psyllid population could help control your psyllid population, and hence the reduction in the spread of the diseases in your estate. So we recommend the IPM program, which balances chemical and biological control, because there are some natural enemies, and so you have to be careful what you spray. So as I said, this is more of an entomology department, and I wouldn't really go any further, but it's just to say that keeping the psyllid population low is critical to reduce the spread. It's one of the means and methods. So you manage the tree health. So you have to have proper fertilization of plants. Now remember, I didn't actually put a fertilizer recommendation here, because you shouldn't do a soil test to get a generalized recommendation. As you've seen before, you could even have any one of those minor nutrients not present in your soil, and it could just simply be that your pH is too high. So we wouldn't give a general recommendation, but we would ask you to contact the research division to get a proper soil test done, and then once that is completed, you can probably ask for a more generalized recommendation, that a more specific recommendation to you. But remember, the fertilizer, it just helps keep the tree alive and make sure that your fruit remains healthy. And doing this along with your spray program will also help you combat the HLB disease. So these are just some general recommendations for the psyllid population. If anybody wants a copy, they can at the end email the email address, and we would forward it to you. So we just want to say that even though HLB is in the country, there are other deadly citrus diseases such as citrus leprosis and citrus canker. So please contact the Ministry of Agriculture if you feel that you have any problems, and this is the ministry's email address. It's ppdiagnostic.laboratory, at gov.tt, right here. So any questions? That's the end of the presentation. I hope it was informative. So I'll just check the Facebook page for any questions. So we have a question. We have some questions. Could you just give us a minute to sort out it? So we just Elizabeth Jones. Oh, with your tree. Could you please? Let me go back to that. Could you please? Yeah. Could you please send the email to? Oh, has a G in it. Hi, sorry, we have an issue. It has a. My colleague just corrected me. It has a G in the email address. Capital, right? So I would read the email again. It's ppdiag capital LABORTORY at gov.tt. Now, as you've seen from the presentation, you said the tree is yellowing, but it could be a nutrition deficiency. So could you please kindly send us some pictures at that email address, and we would respond to you in a timely manner. Dino Mohammed, I think that's her name, but in healthy resistant. No, we said tolerant, not resistant. That's two different terminologies. I am not from the ASD. You'd have to call the ASD Agriculture Services Division. They are the ones that will tell you of the availability of plants. However, those are the rootstock is tolerant. So please give ASD a call and let them, ASD a call, and they would let you know. Or you can call through the county office and let them contact ASD for you. Does it have Sahade Mutilal? Does it have a preventative? Does it have a preventative method? Currently, Trinidad has the disease widespread, so there's no preventative method because the vector is very efficient. So you can use IPM strategy, which is to spray and to fertilize. As I said, the vector loves young flush, so if your citrus tree has started to flush, you can try and spray and use fertilizer. But there's no preventative method, and there's no cure for the disease. How can Duncan Williams? How can I access PCR tests? So currently we take the samples, and if we determine that the sample needs a PCR test, we will conduct the PCR test. But generally, most of the time, the symptoms are visible because the fruit is deformed. And so you can just send a sample to the Research Division Plant Pathology Laboratory, and we will take it from there. If you're not sure what to do, as I said, you can email the ministry, the Plant Pathology's laboratory, and we can follow up with you in a timely manner. That's the kind of thing. So I see there's no more questions in the Facebook chat. You can continue to put questions, or if you're watching this after the live presentation, feel free to email We're stressing the email because our phone system has some issues. So the email, you can send pictures of your email. And remember, it's a plant pathology diagnostic laboratory. So it is not only for citrus. We deal with other crops. So you can send emails with pictures, preferably. We all in the technology zone, so just take a little camera. Try it. We probably should do a presentation on how to take pathology pictures. Try to get a good, I don't know if you all know it, have like a macro setting where you can kind of zoom in so you can see pictures. You can hold it in the light to get a good picture. So because a good picture goes a long way. I see you have 2 comments there. So probably that's one of the other presentations you can do, is to show you all at home how to take pictures, because that is one way of us giving you a quick diagnostics. Yes, can I repeat the email address? It's PPDIAG capital LABORTORY. That's correct. Yeah, I just double checking my colleague to make sure the email address is correct. Yeah, LABORATORY at gov.tt. So I know this is a quite short presentation, but I do want to go back and stress one thing with this disease, is that the ministry is committed to making sure that HLB does not affect livelihoods of our valuable citrus farmers. And if you have any problems with any problems with any diseases in your trees, remember, in your trees, please, I urge you to send an email to PP Diaglab. I just can't pronounce it properly, but diagnostic, because I know that's diagnostic Diaglab. As you see here, so that we can address the matter, because remember, it's a national obligation for us to do pest surveillance and reporting. So I know I've taken some of your lunchtime, and it was a pleasure to do this presentation on HLB. As I said before, we did one on citrus canker, and we're stressing that my colleague Mr. Terence Jack did that presentation, that citrus canker is not in Trinidad, but citrus greening is in Trinidad. So it's 2 different types of program. Citrus canker is we are trying to determine, making sure that it's not in Trinidad and stay out of Trinidad, and citrus greening is in Trinidad, so we want to manage this issue. So even if you have a field and you're not sure, or if you have a plant in your house and you're not sure, please send us an email. We are here to help. So thank you from the Ministry of Agriculture. We'll just give you all 1 to 2 minutes for any additional questions on the Facebook page. If not, and you're too shy to ask one on the Facebook page, please send your email to the ministry's plant pathology, as I said, it's a plant pathology laboratory. So please send pictures that you think something may be wrong with a plant at home, and we will try our best to help you. So we just give you 1 to 2 minutes for any other questions. If not, we will end the presentation here. Okay, well, we are not seeing any presentation, any other questions. Thank you for your attention, and good evening.